All right. We transcribing? We are. Okay, everyone. Hope you're doing well. Hope you had a wonderful spring break. Welcome back. Today is Wednesday, March 25th. This is biotechnology. So I have been slacking behind, guys. I apologize. I'm getting caught up now. So tonight, just a couple announcements here. Tonight, we are doing chapter 8, DNA fingerprinting, which is going to be included for exam 3, which isn't until April 6th. Okay. And most likely tomorrow I will have chapter 9 completed, and then I'll be caught up, and then we'll get chapter 10 to you as well. So yeah, that exam is not too far off. I will get a review sheet to you as well once we get through the chapters. So you'll have it that last week to assist you in studying. And we'll get that done here in just a minute. So also your paper. I've been getting I received some excellent outlines from people before spring break. Remember your outline. There was an outline due last Sunday. Not this last Sunday, I'm sorry, the Sunday before that. Sunday like before spring break. That would have been the 16th, right? So I did have a number of people not submit an outline. Almost a quarter of the class didn't submit it. So if you didn't submit it, the purpose of the outline was for me to give you, a thumbs up or a thumbs down on, what you're accomplishing so far in your paper, how I think your paper is going to be organized, and if it's going to include everything that I was hoping that it would. So, a majority of you did a wonderful job. There were maybe just one or two outlines that I kind of had to provide some feedback on, and that's okay. That's why we do the outlines. So, use those suggestions when you're putting together your final draft. I think it will make a big difference in your grade as long as you're following that. But if you didn't receive any significant feedback, then you are doing a great job. You're on the right track, and stick with it. So if you didn't turn in an outline, though, now you have no feedback. So it's kind of like you're going to be going to Vegas when you turn in this paper, I think. So that being said, I have done two things. So one is I changed the due date for the final draft, and I did that in part because I got the feedback back to you a little late over spring break. I apologize. I did try to take a little break this last week. It's very nice. And so that kind of makes up for that. And then also for the people that didn't submit an outline, I'm giving you another chance to submit it. There will be a penalty to your grade, but I wouldn't worry about that as much as it's an opportunity for you to let you know if you are on the right track with your paper. Okay, so if you didn't turn it in, a good amount of time before the paper is now due. What is that? Did I change it? Yes, April 13th is the new due date for the final paper. So I would say here, try to get it to me by April 6th if you haven't turned one in. But that way I can give you some feedback. I'll get it to you right away. You'll still get some points for it. And, that way you'll know that what you're submitting for your final draft is definitely going to be at least up to snuff in terms of what you're including in organization and things like that. Okay. So, like I said, if you didn't submit an outline, try to get one in by April 6th. It's not necessarily a deadline. You've already missed the deadline, but at least you will get an opportunity to get some feedback. Okay. All right. So we're going to get into chapter 8 here. It's not too long of a chapter, a little interesting. There's a couple people that are doing a research paper on DNA fingerprinting, and let's get to it here. There we go. All right, so this chapter is on DNA fingerprinting and analysis. We're not going to cover all these sections, most of them, but not all of them. I definitely took out the non-human DNA analysis, and I think I put took out some of 8.3 here. All right, so what is the definition of forensic science? I think this is a kind of poor definition, but they're defining it as an intersection of law and science. And that makes sense to me, though. So, it's using science to enter into the legal system, I guess, right? So they gave some examples, though, and I like this, like, photography, definitely the ability to use, the chemistry that's involved in developing film and whatnot, and the physics that's involved in developing optics for photography is definitely, you need to have some scientific background to do that. So that was, I guess, the first example they were entering in, although, I don't know, I swear I've heard examples of other chemistries being used, even as soon as like the late 1700s. I just can't remember a specific example, but to identify things like blood and stuff like that. And then in the early 1900s, fingerprints. And so, the most significant advancement since that has definitely been the DNA fingerprinting, which is what we're going to talk about tonight. Started in 1985. And, this is such an advancement because there's just no room for doubt with DNA fingerprinting. Fingerprints are good, but it can be difficult. It's difficult to collect a fingerprint. It's actually easier to collect DNA than it is a fingerprint, and also there's a significant overlap in fingerprint matches. So with DNA fingerprinting, though, there's just no chance. Well, that's not completely true. There is still a chance that it could be wrong, but it's very slim. Why is it slim? Well, because every person has a unique DNA profile. They have unique sequences within their unique DNA sequences in their cells. We all have the same chemical DNA. It's just the order of the base pairs, the A's, T's, C's, and G's that differ. And to make it even more powerful of a tool, the organization of my base pairs differs, between me and you. But all of the cells in my body have the same profile, right? So however you submit, however you get DNA from an individual, whether it's through saliva, blood, an actual tissue sample, the skin, the hair root, it's the same. Right? There's no difference. The DNA is the same. So 1/10 of 1% of DNA differs from person to person. That's good and bad for DNA fingerprinting. So it's good if you know the places, the loci where the differences occur. If you don't know, then it's bad. I mean, early on when they were looking at sequence alignments, it would be very rare, slim to none, that they would find differences in different people. But as they started to look more closely and get more reliable results, they started to see that there are certain what are called microsatellite regions in people that are consistently different and also consistently the same within all the cells of the individual. So there are two types of tests that are done. One is more prominently done. The two types are RFLP, which stands for restriction fragment length polymorphism. The other is PCR, polymerase chain reaction. So you guys should be familiar with PCR. Now we should be familiar with both of these because we actually covered a little bit on both of them way back in chapter 2. But we're going to go over them both tonight. So restriction fragment length polymorphism. That's a great word, a series of words. So restriction, what they're talking about are restriction enzymes. These are enzymes that cut DNA at specific sequences, restriction enzymes. Those are the enzymes you find in bacteria, protect the bacteria against bacteriophage. And when you cut a piece of DNA, now you make these different length fragments, right? So that's the fragment length that you can run on a gel and see the sizes. Polymorphism is a change in the DNA sequence. So what that means is that that enzyme looks for a particular sequence in the DNA. Let's say it's GATCCA, okay, something like that. And the enzyme, if it finds that sequence, it'll cut the DNA right there, making one piece into two pieces. But if that sequence is changed, let's say it's GGCTTA, then the enzyme won't bind to it and it won't cut it. Simple as that. So if you have the polymorphisms in these specific restriction sites, then you won't cut it. So a lot of these sites have been identified in the human genome. And because of that, RFLP analysis is very accurate, right? So as long as you use the right enzymes that cut at the right locations, you compare those between two individuals, you will see a difference. Now, when I say difference, obviously, you have some genetic similarity to your mother and your father, right? Half of your genome is extremely similar to your mom's, and half is extremely similar to your father's. Remember, you got two copies of the chromosomes, one from mom, one from dad. So, but RFLP and PCR considers that, right? It will show, the DNA that's from your mom and basically the DNA that's from your dad, right? So this is also used in parental typing, trying to determine who the baby mama is, things like that. All right, so the thing with RFLP, though, is you need a lot of DNA to do it, because to do a restriction digest, you can't just have like a picogram of DNA. You have to have a few micrograms of DNA that you cut and then run on a gel. And it has to have really good integrity. So, people that are being undergoing a forensic analysis, if they can, if they're able to get a blood sample from them, or something like that, then you can do RFLP. However, that's not often the case, right? When a detective is looking at a crime scene, they get a very small amount of DNA, maybe just one blood cell, one cell's worth of DNA, not a lot. And it might be completely dehydrated or exposed to some chemical or whatnot. So PCR comes along. It's a much more sensitive test, and it can use a lot lower of amount of DNA. And, as long as the primer sites are generally still intact, you will get amplification. The problem with PCR, though, which you don't run into in RFLP, is PCR is so sensitive that you risk contamination, right? So, and I don't mean just like another person, like, that was there at the crime, but the technicians that are collecting it, the technician that is doing the analysis in the lab, when they do, PCR like this, you have to do it in a sterile cabinet, sterile hands, pipettes, gloves, sterile tubes, all the stuff. And it doesn't only have to be sterile. You're not worried about microbial growth. It's to be DNA free, right? So a lot of ultraviolet lights will be used to sterilize the surfaces and whatnot. Air, recycling the air in the hood, things like that. All right, so just kind of look at this RFLP analysis a little more closely. I don't really like how the author has done this with these double helix things, but basically what do you have? You have a piece of DNA, right? And you're exposing it to a restriction enzyme. And look, it has cut that DNA into three pieces, right? So, and then you have another individual and you give it the same enzyme, and look, it is only cut it into two pieces, right? So, just when you're analyzing these agarose gels, you should know that W plus X plus Y equals individual one, right? So if you add up the sizes of all these, it will equal the size of this piece of DNA here. And if you add up Z and Y here, it would add up to the size of this piece of DNA. Right? What I don't like about this figure is it doesn't really show the restriction sites very well. I think it's a little misleading, but basically you get three pieces here and you get two pieces from this individual. And when you run it on a gel, you can see the three pieces, right? Y's are the smallest, W's the largest in this one, and you only see two fragments here. So that means that there are differences, there are polymorphisms in these individuals, and they're not the same individual. Okay, this is considered a DNA fingerprint here. So just a little more about the polymorphisms that are being looked at. Polymorphism is a very vague term, and this is going to get a little annoying because there's a lot of synonyms out there for all these different types of mutations and natural changes in DNA. But polymorphism is a very, very general term. It's any change in the DNA sequence. Okay, so there's another term which looks at, it kind of zooms in the genome of the areas that we're looking at, which are called variable number tandem repeats, right? So what does tandem mean? Tandem means like one after another after another. And repeats means that one after another is being repeated. It's the same thing that's one after another. It's immediately after another, after each other. So the repeats, like if your repeating sequence is ATT, you're going to see ATT, 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 and they're all going to be clustered together. These are VNTRs, variable number tandem repeats, and they can vary all throughout the genome. That ATT example you can also have one that's like 50 base pairs long, and that repeats over and over again. So your VNTRs, my computer is saying it wants to restart. Don't do that. So your VNTRs are unique. There's a lot of events that occur in the cell. Not only just random mutations, but things like crossing over and chromosomal translocations. And VNTRs are unique. They're unique to your, even within your lineage, within your family. So another reason looking at these repeating segments is a good idea is for privacy reasons. So obviously, FBI has a data bank of as many people it can gather of all their DNA, and it generates a profile. That way, anytime a crime is committed, they don't know who did it. They have a DNA sample. They can compare it against their database of, I'm sure what now is millions of individuals. But if anyone got their hands on that data, it would be, it wouldn't be useless, but it would not invade your privacy all that much, right? It's not going to be used to, it couldn't be used to like deny you for insurance or something like that, because these are just repeating sequences. They don't provide any information about your proteins, about how your proteins. So they can't be used to identify disease or anything like that. It's just a repeating frequency. So because they're unique, because they don't provide a lot of information about the individual, just that they're unique, it's very good as a scientific marker for identity. All right, so these VNTRs, there are a bunch of different types. Right, the specific type that forensic scientists use are what are called microsatellites, and the synonym for that is short tandem repeat. Okay, these are types of VNTRs. And what I mean by short is like between 1 and I think 8 is about the 1 and 6 base pairs is about the limit that it goes. So if you look at this repeat here, this is an AAT that's repeating, and you've got it repeating about 8 times. Okay, so you've got two things to consider here. You've got the number of repeats, and you've got the number of base pairs that are repeating. Short tandem repeats are just a few base pairs, like between 1 and 6 base pairs. Okay, so this repeat is unique in this area of the chromosome in this individual. So you have to think about it, right? And we can use PCR to get this sequence here by generating primers to these sites, right? To this site that is upstream of the STR and downstream of the STR. Right? Remember, primer is an important component of PCR. All right, so what sites have been identified that provide good information to generate a DNA profile? Right. So currently there are 13 of them, but I thought they're adding new ones I think they're adding new ones here soon, or they've already added. I don't really know. But there's 13 sites. So remember, these are 13 sites, 13 loci, right? Locations of the chromosome. Here's one location, here's another location, here's another location, here's another location. And these three here are all in a very similar location, right? So this is considered one loci. That's why if you count these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. I counted 15 here. That's not 13. So you should group these together. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, right? So 13 unique loci, all right? All this data is uploaded. It's all put into this combined DNA index system called CODIS. And if you get interested in this, we're going to refer to this a couple times. Here is the National Institute of Standards and Technology. This is the Department of Commerce, but it goes through all of these standards that are utilized in CODIS, right? So you can peruse this if you're interested in looking at these different, but it's, all this data ultimately comes from GenBank, which is through the NIH Department of Health. Okay. All right, so let's look at specimen collection. So the thing with specimen collection is contamination, especially using the PCR, you have to consider contamination. So how retentive do these people get that are going out and collecting samples at a crime scene? Extremely. They most of them have to wear protective. I mean, nowadays I've heard of people wearing these just entire suits, so the DNA evidence is not given any scrutiny when going to court. But you have to be masked, disposable gloves, even disposable instruments. No talking, no sneezing. Everything is video. The whole process is video recorded, and they have to be kept dry, right? So there's no mold, things like that. So a lot of training and a lot of standardization, regulation has to be put into collecting these specimens, so it's done right. They have to be preserved correctly in the correct buffer and detergent, right? And they have to get free of contaminating organisms, moisture, sunlight, high temperature. All these things can degrade or contaminate the DNA. So ultimately, DNA fingerprinting, though, is termed a comparative process, right? You have to compare your results to other people. So, a detective gets a sample, they run it through the forensics lab, they generate a profile, and now they run that profile against the database that the FBI has, which I'm sure is many, many individuals. If there's no hit, then they keep going, they keep looking. So they keep, they're screening people that have had something to do with the crime or whatnot. When they're collecting samples from people, the best sample that you can collect is a blood sample, because the white blood cells in there can be easily extracted, and they contain a lot of DNA. You got full cells that have their DNA in it. It's a lot easier than collecting it from tissue, because then you have to homogenize the tissue. You've got all the collagen in there and all that stuff. Getting it from, basophils, eosinophils, it's a lot easier to do. Okay, so now you've got your cells from the individual. You do a little purification, get rid of the erythrocytes and keep the leukocytes, and you bust them open and you treat them with sodium hydroxide and things like that to purify out the DNA. Run it on a filter, and you retrieve it, and now you're ready to do PCR. Okay. All right. So, how can you purify DNA? Chemically, it's the most common. It's a 3-step process. You treat with sodium hydroxide. You then treat with sodium acetate, and then you treat with sodium what is the third one? Ethanol, right? So sodium hydroxide, sodium acetate, and ethanol. So the sodium hydroxide is a lysine agent, like it breaks open the cells. The sodium acetate helps bulk up and bind the DNA. And then you stick that on a column. It's nice because DNA has a negative charge, so it will stick to a silica column. And then you flush it with ethanol. The ethanol will wash out all the protein. And it will get another restart thing here. Postpone restart. Sorry. This computer. So you flush it with ethanol, and that'll pull out the proteins and anything. You just have DNA. All right. So, and then the work that the technician does, I will tell you, is very simple. Like, it's really just like you get companies. Amersham, for example, sells a forensic kit that it's got the tubes, and you just take your DNA sample and you mix so many microliters of these different tubes, which is like a mosaic of all the primers that are used for the amplification. As long as you have good training, it's a relatively simple process, and that's good because that means that there's less of a chance for error. All right, so the PCR, then you're ready to process your DNA, right? So PCR, you're going to amplify those STRs, right? So you use primers to bind to sites that are flanking the STR, and then you get amplification. Right? Primers are about 18 to 22 nucleotides long, and they're complementary to the regions that flank the area of interest. And after you have your primers and your DNA polymerase, your buffer, your DNTPs, you've got them all in a tube. You throw it into that machine that is a thermal cycler. And now you have amplified this region of DNA millions of times. Okay. And it's enough to see on a gel or to sequence. And you can get a profile of that. Why do I have this here? No, just reiterating. These are the sites that are amplified. So, every DNA, you think about it, every DNA profile reaction will have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 13 times 2, 26 primers in it. And that's not including any controls that I'm sure that are run. All right. So, yeah, PCR is your DNA. Right? We're interested probably of what's in here. So we generate primers, which are complementary sequences to the DNA. And then the steps in the PCR reaction are denaturation, right? The DNA comes apart, becomes available to the primers. Then annealing, which is the primers stick to the DNA. And then the extension, which the DNA polymerase does, it binds to the primer and then it copies the DNA. All those are mediated by different temperatures. So it's a thermal cycler. It's cycling those temperatures from 95 to anywhere from 55 to 62. And then finally the extension is 72. And now you have, exponential growth of your numbers of DNA. All right. So after amplification, they're put through electrophoresis. Now, what we're familiar with is the electrophoresis that looks like this, just simple agarose gel electrophoresis. And when you're doing RFLP, you can do that. But PCR uses a little variation of the electrophoresis. It's the same principle. It's just done in a capillary. So you use these small capillaries. And then as the DNA comes out of the capillary, it's analyzed by like a kind of a spectrophotometer type thing. And it will give you information about it. It will give you the length of the fragment very precisely, right? And it will also give you the quantity of the DNA and whatnot. And then, so the shift here, like as you go from right to left. So what you've got here, let's take a look at this figure here. Does this have all 13? It does. This would be a complete, no, DS818. Yeah, it does. It's got all 13. Okay, so why are there 2 peaks per loci, right? So this was a in-person class. I would be asking you that and tapping the desk, waiting for an answer. Why do you see 2 peaks per loci. Well, remember, one chromosome is from mom, one chromosome is from dad. So one of these are from mom, and one of these are from dad. Still getting this thing here. This one's from mom, this one's from dad. Okay, but so if you were to run mom's here, and we'll have an example of that in a little bit, she would have either this peak or this peak for this one. She would have either this peak or this one for this one. Okay. This is a report on the amount of DNA that was run. So 500 micrograms was run. It's a lot. And that's it. All right. So this is capillary electrophoresis. Okay. So when the terminology that's used, they call these different peaks alleles, right? So one allele you get from your mom, one allele from your dad, which works in terms of genetics as well. What is this here? Okay, so any of these can be looked up on that NIST, the Standards and Technology site. So I just want to kind of walk you through one of those. So let's look at the D7S820 STR loci. Okay, what I've done is I pulled it up for you on NIST. So here it is, D78S820. Okay, the author's given us some information about it. It exists on chromosome 7. It contains between 5 and 16 repeats of the GATA sequence. Right. So look at this, chromosome 7, and then here's our repeat number. It contains anywhere between 5 and 16 repeats of GATA. Here's GATA 9. Here's GATA 10, so forth. Okay. So, which one are you? Well, you'd have to get your profile done, but, on chromosome 7, right, with this location, right, Q arm at location 21.11, you have a short tandem repeat that exists. It's either between 5 or 16, but it's unique to you. Okay. And you have 12 other of these that can be used to really identify you with a lot of statistical power. Right? That's what I'm trying to get at here. So let's look at another one. Individual D7S820 was 10 and 15. What if it inherited a copy of 10 repeats from one parent and 15 repeats from the other parent? Let's look on the previous figure. So D7820, right, so that's this one here, right? So there's one from mom, one from dad, and so 10 and 15 repeats. All right, so let's say 10 repeats of GATA, right? 15 repeats of GATA. So one allele from mom and one allele from dad. Okay. There are 12 alleles for this STR and 78 different possible genotypes. 12 alleles. Let's see if that matches what's here. So that's not what I've got here. Well, actually, no, this is an allele. So 1, 2, just curious, 1, 2, 3, 4, 5, 6, 7, 8, 9. No, that doesn't work. I don't know. That might be a little outdated or something, because what I see is all of these alleles. These are all possible alleles here. Okay. Anyways. All right. So now you have the profile, right? You have something that looks like this for an individual. Sorry. Something that looks like this. And how do you make that useful? Well, you have to compare it. All right. And with it's like very polite of the FBI, but what they do is they make their comparisons based on exclusion. Okay. So, like I said, if let's say they are looking for a match, right, at a crime scene, and they get your sample, and they say, "Oh my God, this is the same blood that of you were at the crime scene, right? So you were there." That's not the end of the story, though. That's in terms of forensic DNA testing. They see that you were there. Now they're like, okay, well, we're going to take the sample that we found at the crime scene and your sample, and we're going to test it against a whole lot of people in the population. And that's why they have their database. And they do that to like show in court, like, hey, we've taken this person's sample, we've taken the DNA that we covered at the crime scene, and they're the same, and we've checked it against 50,000 other individuals, and none of those 50,000 other people have the same profile as John Doe here, right? Gives it a little more power in terms of court. Okay, so for example, here we've got some different, like a case mock-up. We got Jane Doe, we got Dorothy Smith, right? And we have 2 stains on the knife. Okay, so knife blade A, stain A. So this is the blood that they recovered. Now, if you look, so I'm just like scanning, I'm comparing stain A and stain B. It has the same profile, right? There's no difference. No difference. So we don't have to really look at both of those. We can just look at any one. And we take that, and now we compare it to the individuals, right? So the knife stain is the same as Jane Doe there. Look, 1516, 1516, 1619, 1619, and so on. Okay? It doesn't say that she did it. It doesn't, like, you couldn't say she did it and use this in court yet. But what we can say is Dorothy Smith did not do it because she has different profile. You can see there's still similarities though, right? 16 and 16. You're not going to get a same or different in every single one necessarily, right? There is going to be some similarities between you and the population, but when you look at all of the loci in the end, cumulatively, it should be there should be differences in there. And that's where the statistics come involved, and we're not going to get into that. That's beyond the scope of this. But so this says that Dorothy Smith can be excluded. She did not contribute to the DNA found on the knife. Okay, and then you'd have to start testing more people and making sure everybody else gets excluded. Maybe test Jane Doe again, even make sure that contamination didn't get in there, and the technician was the one whose blood was on the knife. But it is a science of exclusion. All right, so I thought I took this out. Oh, that's right. Okay, so first case that was ever used was this murder case, and so it was a sexual assault, murder of a girl. This is the United Kingdom, and that's right. The suspect's DNA did not match what was found at the crime scene. So how did that happen? Well, police collected 5,500 samples from the area. None of the profiles matched. Right, it's really scratching their heads there. And they knew that they had the suspect in that group, right? But they just couldn't get a match. So why was that? Well, that's because a friend of the suspect had given a sample in the name of the suspect, right? So there was like a thing with forgery and of the identity documents and stuff. And I even heard, I was watching one of those crime shows, and there was a surgeon who I think offed his wife, and he took a vein from a pig, and he surgically implanted it in his arm, filled it with human blood of another individual, and when he go in to give his went in to give his DNA, the technician would stab the vein with a pig vein, and it obviously wouldn't test positive, wouldn't be the same as what they were collecting from the crime scene. The detectives started to suspect what was going on. They had him redo the test. I think it was like multiple times until they finally figured out what was going on. The surgeon was so good at getting this in and leaving no scoring and whatnot. But I tell you, man, there's some wacky people out there. anyways, all right. So just something that's kind of unique when you're isolating. So I guess it's common to have to be able to distinguish between sperm DNA and vaginal DNA from a mixed source, obviously for obvious reasons. Why can that be done? Well, so sperm DNA, so the spermatozoan, right, are they have a protective coat on them, and you can distinguish them chemically through this process, right? So basically, you would take a vaginal sample, which would have the vaginal cells and possibly sperm that was in there from possibly a criminal, right? So you mix it up, and then you put a buffer, and then you centrifuge it, and all that stuff is down there, right? And then you use this chemical called DTT. So DTT is it's like a denaturant, right? Very powerful denaturant. It'll even break up like chess, break up proteins and whatnot. And so you put it in there, you mix it in there, and it won't. It'll break up the cells, right? But it won't break up the sperm. The sperm are protected from it. So you collect after you add the DTT to the specimens, you have vaginal DNA in the supernatant, but the sperm are still intact. So you collect some supernatant, put it aside, and you have the vaginal cells. Now you still have the sperm in there, and you can blast those open with either high heat or sonication, and now extract the DNA that's in the sperm. So you've selectively isolated sperm and vaginal DNA. So there's a lot of techniques that are used to pull samples out, especially if you have like a pool of a bunch of individuals' DNA and you're trying to sort through it. There are techniques that you can do to sort through that as well. Forensic profiling is also used in paternity testing, right? So I always remember that. You guys might be too young for that. That Maury Povich show. You are not the father, and the guy does a dance. But so what's unique with paternity testing, the PCR can be used. So like, look at this. You got a paternity dispute, right? So this is mom. This is the alleged father. I like how they call him the alleged father, like it's some crime, and the child. So let's look at the child's STRs, right? So he's got a peak right there that lines up with mommy, right? And he's got a peak there that lines up with daddy, right? So this peak, so the child's peaks, all of them should be present in either the mother or the father. This peak here lines up with mommy, right? This peak here lines up with dad. Okay, dad has his own that he got from his mom or dad that didn't pass down. Oh, sorry, mom here has his own. And this child, this peak here lines up with mommy. Oops, sorry. And this peak here lines up with dad. Okay, so this paternity dispute is no longer in dispute. Obviously, half of the chromosomes of the child are from the alleged father, right? So this is the way it's done. But if if you can't analyze by RFLP or STR, right, such as samples that are too old or you just lack cellular nuclei, then you can use mitochondrial DNA. So, mitochondria is kind of unique. So mitochondria are they're organelle in the cell that provides ATP for the cell, right? The energy for the cell. And all your cells have it because all cells need ATP. And when you look in terms of like evolution, the mitochondria came from bacteria, right? So what happened is like a cell that didn't do aerobic metabolism, so an anaerobic cell, ended up engulfing an aerobic cell, bacteria. And that aerobic bacteria over millions of years became the mitochondria. And that aerobic bacteria, which became the mitochondria, retained its genome, its circular plasma or circular genome, piece of DNA, its chromosome. And that is our mitochondrial DNA. It's reminiscent from millions of years ago of evolution of from a bacteria. Now, during fertilization, human fertilization, all right, the egg gets contacted by the sperm, and what happens is it contacts the coat. There's a tip of the sperm is called the acrosome, and it contacts the surface of the egg, and the acrosome dissolves, and it spills out its genetic content inside the egg, and that's fertilization. So the mitochondria are not located in the tip of the sperm right there. They're actually located further to the back to provide energy for the little tail, right? So that flagella needs a lot of ATP. So during fertilization, the mitochondria that are in the egg, which are the mother's mitochondria, remain, and that when that fertilized egg or zygote divides, those mitochondria split off to the two daughter cells. But dad's mitochondria didn't come in. So all of your mitochondrial DNA is like your mom's, right? And all of her mitochondrial DNA is like her mom's, and back, all the way to the caveman. It goes all the way back. Yeah, so none of your mitochondrial DNA is like your dad's. Now, is that important are you more like your mom genetically than your dad? No, because those mitochondrial diseases, mitochondrial genes, they're very similar in males and females, and there's not too many. They just have to do with the mitochondria. They provide the codes for the ribosome, which you need ribosomes inside the mitochondria to make these proteins that are used in the electron transport chain. Cytochrome B and nicotinamide adenine dinucleotide dehydrogenase, right? These chemicals are important in generating ATP. But you can see here, so like here's mom, mitochondrial DNA from mom, mitochondrial DNA from another woman, and it has a different profile. There are different STRs in that. But the mitochondrial DNA that's passed in families, so here's mom, every child has mom's mitochondria, and every child has mom's mitochondria, just like that. So these children have this person's mitochondria. Okay, if you're doing STR, it's very useful in paternity too. But you only do this sort of analysis if you can't get genomic DNA. And I just want to make a correction. Are you more like your mom genetically than your dad? Yes, technically, because the X chromosome is larger because you have whatever. Anyways. All right, that's all I got for you guys. I hope you have a great night and or day, whenever you're watching this, if you're watching it. I will have chapter 9 out very soon, tomorrow or the next day, to get caught up. Okay, so I hope you guys are doing well. Work hard on those papers. Stay up on this information. Read through the chapter, work the questions at the end of the chapter. Reach out if you need any help, and do your best. Study hard. Do well in school. Talk to you soon.